As director of the Comprehensive Memory Center in the Goldrich Institute, Dr. Okhravi has created an amazing model of care that includes a comprehensive program for assessment and management of patients with cognitive disorders, as well as working with their caregivers. The clinic receives support from multiple philanthropic sources and has a robust research infrastructure for clinical trials and other types of research studies as part of our efforts to better understand this dreaded disease from the patient and caregiver perspectives, in the hope that someday soon we can eradicate this devastating disease, and which affects nearly 7 million people in the United States. By the way, excuse me, it's Older Americans Month, and next month is Alzheimer's Awareness Month. So our program today is timely, as those most people impacted by Alzheimer's are over the age of 65. Many thanks again to all of you for participating today. And also a friendly reminder, please remain on mute. If you have any questions, please feel free to post them on the chat. Now, please help me welcome Dr. Okhravi.
Thank you very much, Dr. Castillo, for the nice introduction and beginning of our fifth educational program. This is just so exciting. Let me see if I can share my... Okay, share screen. Okay, so this is my screen.
Looks good on our end.
Great, wonderful, wonderful. I have really to say that, within the past 2 decades, I've been deeply involved in the field of Alzheimer's disease. And let me tell you, there has never been a more thrilling time to be a cognitive disorder specialist. The recent advancements have really sparked lots of excitement in the field, especially in the field of therapeutics. It is really wonderful to witness these breakthroughs and be part of this journey. So let me share part of that excitement with you today. That's what I would like to do. So as you can see, I'm going to talk about the most recent advancements in the therapeutic field. Let's see if I can forward my slide. So the overview of my talk, first, I will talk a little bit about Alzheimer's disease as a biological process. And following that, I will move forward to talk a little bit about different treatment strategies, symptomatic versus disease modifying. That is true for any type of disease processes. Then I'll talk a little bit about the clinical trials within the last 20 years, both the failures and successes, and mainly that's actually failures. And then the main part of my discussion is about second-generation anti-amyloid monoclonal antibodies, and those are 3: Aducanumab, Lecanemab, and Dunalimab. I will share the results of these 3 antibodies, the medications, the research, the clinical trial with you. And then I'll touch base on prevention trials, mainly talk a little bit about the AHEAD study, and also lay out the plan for future of clinical trials. So that will be the overview of my talk. So let's start with the Alzheimer's disease. Alzheimer's disease neuropathological hallmark, or the culprit of Alzheimer's, has not changed since 1906, when Dr. Alzheimer introduced the first case of Alzheimer's to the medical community. That is essentially 2 different culprits. What you see right now on the left-hand side, that's plaques. Let me see if I can use also my pen here. So it's mainly plaques, which is made up of amyloid, and you can see that here. and also tangles. You can see tangles here. And tangles are, neurofibrillary tangles are made up of tau protein. And what we think that the amyloid protein, the amyloid plaques start a cascade of events that they will lead to development of these tau proteins and tangles. And tangles are toxic to our nerve cells, and they can cause damage. How do we diagnose right now in the 21st century Alzheimer's disease? There are two very powerful tests that we use. One of them is spinal tap, which is very accurate. And the second one is a type of PET imaging, a type of scan, which is called amyloid PET imaging. And what you see here is the amyloid PET imaging from two of our patients. On the left-hand side, that's from one of our patients who doesn't have Alzheimer's, and the right-hand side, that's a patient who has Alzheimer's. These cuts are horizontal cuts, and you're looking from the top. What you see on the right-hand side in hot metallic color here, especially in the rim, in the red, that's amyloid plaques. Those are amyloid deposition in the brain. What you don't see on the left-hand side is that hot metallic color in the rim, again, the thinking part of the brain. So essentially, for this patient on the left-hand side, that's negative amyloid imaging, meaning that this patient doesn't have Alzheimer's. On the right-hand side, that means that this patient has Alzheimer's disease. This test is very accurate. It's 95% accuracy. That's quite high. But unfortunately, it's quite expensive. It's about $10,000 here in our town, and it's not covered by Medicare or other insurers. But we do have, of course, available research studies, and some of them are here, a head study, new ideas, rephrase, that we can order that for our patients free of charge. But of course, that depends whether patients will be eligible for those studies or not. So this is the way that we diagnose Alzheimer's quite accurately. Now, there are different hypotheses about how Alzheimer's disease develop. And one of the most popular one is the amyloid cascade hypothesis. And that's like kind of this fall that you observe here. Based on that hypothesis, the amyloid protein is very actually sticky. They stick together, become 2, 4, 10, 100, 1,000, and thousands. And then they become plaques, as you can see here. And as I mentioned before, these plaques can trigger a cascade of events that will lead to production of tau or neurofibrillary tangles. And when neurofibrillary tangles develop, those are toxic to nerve cells, and they will cause nerve cell damage and neuronal cell death. And as a result, people may develop symptoms of dementia and Alzheimer's disease. Now, based on this hypothesis, if there is any way that we can prevent or remove those plaques from the brain, we should be able to stop production or reduce the production of tangles or tau. And as a result, we'll be able to slow down memory loss or even prevent that. So essentially, these medications that I'm going to talk to you today, they put this hypothesis on trial. And let's see if that is true or not. There are 3 medications, as you can see here, that those are either FDA approved or they have designation of breakthrough therapy. The first one on the left-hand side, as you can see, that's Aducanumab. Aducanumab received FDA approval on June 7th, 2021, almost a couple of years ago. Lecanemab got accelerated approval January 6th, 2023, just this year, earlier this year. And Dunalimab, that they released the results just a few days ago, I think that was on the 3rd of May, which is quite exciting. That also was positive, but that medicine right now has FDA breakthrough therapy designation, but they are going to publish the results in July. And after that, they will go before FDA to get the approval. So these three medications are the medicines that I'm going to discuss with you. But first, let's see, what is it, breakthrough therapy designation? What does that mean when we say breakthrough therapy designation? I'm going to read exactly what it says. These medications aim to expedite the development of drugs that are intended to treat a serious condition like Alzheimer's when preliminary clinical evidence indicates it has substantial improvement on an endpoint or endpoints over already FDA-approved medication that right now we have available. Let's talk a little bit about different therapeutic approaches that we have available for any disease processes. What you have here on the horizontal line, that is age or time. And what you have on the vertical line, that's the cognitive and function, and the arrow shows the direction of decline. So this is about Alzheimer's disease. In solid blue, that's the natural history of Alzheimer's disease. If we don't really give any medication or anything, people will decline cognitively and, of course, functionally, and at a certain point, they will die at this point here. However, if we have symptomatic treatments, we can give them a little bit of an oomph and improve their symptoms, but still, because these are symptomatic treatments, the outcome and the result will be the same, and we won't really change the trajectory. As you can see, they end up kind of almost the same timeline. All the medication, traditional medications that we've had so far, those are symptomatic therapies that we've been using within the last 20, 25 years. And those are donepezil, rivastigmine, galantamine, and memantine. Those are all symptomatic treatment. What we really like to have in the field is disease-modifying therapy, as you see here in green. And that means that medications that they can attack the underlying cause of Alzheimer's, for example, amyloid or tau, so we can change the disease trajectory, so we can slow the slope of cognitive decline. Those are the medications that really make the difference. Symptomatic therapy is very important, but we really need to have disease-modifying therapy, and we haven't had, but most recent medications, they have proven to be effective, and they are disease-modifying therapy. So, 3 medications that are considered as disease-modifying therapy: Aducanumab, Lecanemab, and Dunalimab. Let's talk about or review the Alzheimer's disease drug development pipeline from 2002 to 2012 in 10 years. I just want to give you a little bit of a background. I warn you, this is a very blue and sad and depressing slide, but this is the reality. Within 10 years, from 2002 to 2012, we had only 413 Alzheimer's disease clinical trials, not really enough for such a problem like Alzheimer's disease. Total of 244 unique compounds. 36% of those trials were symptomatic treatments. 53% were disease-modifying agents. Overall success rate of these medications for all of those 413 AD clinical trials was only 4.4% success. 99.6% failure. That's quite astonishing. So if you compare to a 20% success rate for a new cancer drug, this was really, I would say, a disaster. If you take a look at FDA-approved medication between 1990 to 2017, almost 27 years, there were over 1,100 FDA-approved medications, only 4 of them for Alzheimer's disease. But things have changed. Now, this is about Alzheimer's disease drug pipeline in 2022, last year. This is a busy slide, but the reason that I wanted to show it to you is to see where we stand right now in terms of disease-modifying versus symptomatic treatment. Now, here in this slide, the outer circle is phase 1. And phase 1 clinical trials, those are the trials that is mainly for safety, small trials for a short period of time. The middle circle are phase 2 clinical trial, as you can see here. And these are both for efficacy and safety. And when studies move from phase 1 to phase 2 and they are effective, then they go to phase 3 in the inner circle. And phase 3 clinical trials are mainly again for efficacy and also safety, and these are big trials. And if they show positivity in their results, then the sponsors or pharmaceutical companies, they go before FDA to get the approval. What you see on the outer part, it actually tells what type of mechanism. For example, here it says disease-modifying biologicals and disease-modifying small molecules. If you take a look at these studies, two-thirds of them are disease-modifying and 1/3 symptomatic. And that's a good combination. That's what we really like to have. Lots of disease-modifying therapies, and of course, they come from these clinical trials. Now, let's talk about the second-generation anti-amyloid monoclonal antibodies, the ones that we just kind of mentioned very briefly. Well, there is a common theme in all of these trials that I'm going to share with you. First of all, they are all the best type of clinical trials, double-blinded, randomized controlled trials. It means that neither investigators like me nor patients, they know whether they are on medication or dummy medicine. They're all multi-center, multinational, and the population is early stage Alzheimer's disease, people who have very mild memory problem, we call it mild cognitive impairment, MCI, or mild Alzheimer's dementia. And all of these people had positive brain amyloid deposition proven by either amyloid PET imaging or spinal tap. Let's talk about Aducanumab. There was a study called PRIME, and that was a phase 2 clinical trial for Aducanumab. It was finished in 2015, and that showed that the results was positive. And after that, Biogen, who has this medicine, they went ahead for 2 big phase 3 clinical trials, Emerge and Engage. Each of these trials, they had over 1,600 participants. And during the study, after a couple of years, or maybe 3 years, they did a feasibility study. And I'm sure that Biogen wishes that they never did, but they did a feasibility study. And based on that, they decided to stop both of these studies prematurely. But when they looked at these 2 studies, the results, this is what they found. They found that in the Emerge trial, that study showed that the medicine slowed down the memory loss by 22%. So what you see here, this line is the placebo, and this line is the highest dose of the medicine. You can see the difference between these 2 lines, and that was quite significant. Essentially, it showed that there is 22% slowing in memory decline. However, on the negative study, Engage trial, they could not see any difference. That was a failed study. So based on these results, Biogen went before FDA, and they received the accelerated FDA approval, meaning that Biogen needs to run additional study, a phase 4 clinical trial, to show that the results of the Emerge trial, the positive study, is reproducible. And then after that, they will receive the full approval. Now, we are a site for this study, and we will start enrolling participants, I think, within the next month or so. In fact, yesterday, we had our site visit by the sponsor. So this is the results of the Aducanumab. One of them showed 22% slowing. The other one was a failed study. How about Lecanemab? Well, Lecanemab also had a phase 2 clinical trial, very successful phase 2 clinical trial, showed efficacy. And after that, Eisai, decided to go with the phase 3 clinical trial called ClarityAd. This one was also randomized double-blind controlled trial. They had close to 1,800 participants divided in two groups. Treatment group, they were on lecanemab infusion every 2 weeks. And the placebo, they were on, of course, placebo every 2 weeks. Of course, no one knew, neither the investigators nor patients, whether they are on placebo or medication. The duration was 18 months. The median age was 72, and 52% of participants were female. There were some unique features for the ClarityAd. This one was one of the largest clinical trials, more than 1,700 participants in the study. They also had a broad age range from 50 to 90. They included quite broad age range. Most of studies, when you take a look, those are between 65 to 85. But here we had much younger people, and we had also much older people, up to 90, very close to what we see in reality in the, I would say, community. This is one of the most diverse clinical trials. There was 27% underrepresentation compared to average of 4.5 or 5% in other studies. So that was pretty good in terms of underrepresented population. They also included high level of chronic medical conditions. And that's very important because that makes this study, the results, much, much more generalizable. This is the first study ever in Alzheimer's disease showing robust positive outcomes in all of preset outcomes that the study sponsor and the investigators set. Usually they have like 4 or 5, and they try to reach those outcomes. So this was the first one in history of Alzheimer's disease within the disease-modifying agent field. Here's just to show you some demographics. What you see here on the right-hand side, that's actually United States. And when you take a look, you can see the average age of 73. And again, 52% of participants were female. Only 4.5% were African-American. So they didn't do a good job in African-American population, but they did a good job in Hispanics or Latinos by including 22.5% of them. So that was pretty powerful. This is about chronic medical conditions. As you can see here, for hypertension, 2/3 of participants in US, they had high blood pressure. Almost 3 out of 4 had high cholesterol. Almost 20% of them had heart problem or had diabetes, and about 1 in 4 were obese. So those are quite significant, and usually these people are eliminated from other studies. The other interesting thing is that when you take a look at like 2 chronic medical conditions, again, 2/3 of our participants, they had at least 2 chronic medical conditions, and 1/3 of them had 3 chronic medical conditions. One more important factor, which was very interesting, is that they also included people who are on blood thinners. About 6% of participants in US, they were on blood thinners. And this is quite important because these medications can increase the risk of course, bleeding. So they wanted to see if there is any increased risk in those patients who were on blood thinners. This is the results of the study. By the way, these are anti-amyloid medications. What they do, they go to the brain. These medications go to our brain and then bind to those plaques, and our brain will clean it up. And these are quite powerful medications. So let's take a look at this medicine, how powerful it was in removing amyloid plaques from the brain. As you can see here, on the top line here, that's the placebo. And in green, that line is medication. You can see even at 3 months, there was significant amount of reduction in the amount of amyloid plaques in the brain. By 18 months, majority of people, they had no detectable amyloid plaques in the brain. So this is a very, I would say, powerful medicine. Almost 70% of people were negative in terms of amyloid plaques. But this is not enough, right? Because we need to see if clinically these are helpful, if they can slow down memory decline. So let's take a look at the results. But before that, I just wanted to show you, this is one of the participants, and here is before treatment, and then here is after treatment, after 18 months of treatments. And what you can see on the left-hand side, that hot metallic color in the rim, that's amyloid deposition. When you compare it to 18 months later, you don't see any red area in the rim, which is the cortex that I'm right now painting. So essentially all of that amyloid plaques were removed. What you see in the middle here, that's normal, that's physiologic. So this patient on the right-hand side, that patient doesn't have any detectable amyloid after 18 months. So now let's see if this reduction and removal of amyloid was clinically meaningful. So here, this is the same test that they had in IMMERSE trial, the positive trial for Biogen that I just showed. That's called CDR sum of boxes. In green, those are people on medication, on Aducanumab. And in black, those are people who are on placebo. As you can see, at 18 months, there was 27% reduction or slowing of memory loss, people who are on lecanemab. Remember, that was actually 22% with Aducanumab, and now this one is a little bit stronger results, 27%. Now, this is their primary outcome, their major outcome. But studies, as I said, they have like secondary outcomes as well. And here, there were 4 different outcomes. I want to just read to you those outcomes, the results of those outcomes, just to see the numbers. Again, this is the one that I showed, 27%. Here is 26% slowing down. Here is 24% slowing down. And here is 37% slowing down. And 37%, this is about function. So as you see, all of those outcomes, either primary or secondary, they showed that there is slowing down in the memory and also functional decline. Again, quite significant. Now, you may say, okay, what does it mean really 27% reduction? So in clinical terms, how can I really explain that? Well, here in this slide, just to explain what that means. So again, as you can see here in green, that's people on lecanemab. In black, people on placebo. People who were on lecanemab, they had 5.3 months gain in 18 months. What does that mean? I'll give you an example. If I am in this trial and if I'm on placebo, and I'm able to drive my car at the beginning of trial, and at 13 months, I'm not able to drive my car, being on lecanemab will allow me to drive my car for additional 5.3 months. And as you can see, these lines, they still continue to diverge. Of course, this part that is in dotted lines, this is speculation. We don't have data on that. But if that's the case, probably in 25.5 months, we will gain 7.5 months. And probably in 36 months, we will gain more. But that's something that remains to be proven. So lecanemab received FDA approval with accelerated approval designation. What does that mean? FDA may approve drugs for serious conditions like Alzheimer's. Well, there is a medical need, and a drug is shown to have an effect on removing amyloid plaques that is likely to predict a clinical benefit to patients. So essentially right now, FDA is looking at the data, and the full approval probably will come in July, sometime in July. And after that, Medicare probably is going to review whether they cover the cost or not. Medicare categorically has announced that they are not going to cover the cost for these type of medications, but we are hopeful that they may change their decision, and hopefully this medicine will be covered. How much does it cost? $26,500 just for the infusion. Right now, we don't know in the future whether Medicare or other insurers, they cover the cost for MRIs, because there are a few MRIs that people need to have, and also infusion. And the cost for Aducanumab is $28,000. So now the third one, which is Dunalimab, and they just touched base on the results more recently. I put the phase 2 clinical trial of this study. It's very, very interesting. The design of this study is quite interesting, and I think has lots of implications. So for that reason, I put the phase 2 clinical trial for this study. This is kind of a little bit complicated, but let me see if I can explain. So in this study, that the phase 2, they had 251 participants. They divided them in 2 different groups. And by the way, these are all people who have amyloid deposition in the brain, amyloid plaques, either proven by CSF, spinal tap, cerebrospinal fluid by spinal tap, or by amyloid PET imaging. What you see here down there, 126 people, that's the placebo group. And then top 131, that's the intervention, people who were on Dunalimab. If you take a look at people on Dunalimab, they started them on 3 doses of 700 milligram every 4 weeks. So essentially they gave them 700 milligram every 4 weeks for 3 doses. And then after that, they up titrated the dose to 1400 milligram. Now, what they did interesting here, they didn't continue everyone on medication. So the placebo, of course, they were on medication, but in the treatment group at week 24 or 6 months here, they repeated the amyloid PET imaging. The amyloid PET imaging, the way that we read quantitatively, there's something called centiloid. So if readings are under 25 centiloids, that means that amyloid imaging is negative. It means that that patient doesn't have amyloid plaques in the brain. If it's over 25, that's positive. So at week 24, 6 months, they repeated the amyloid PET imaging. For those people whose amyloid imaging showed less than 11 centiloid, way less than positive threshold, which is 25, they moved them to placebo without letting them know. And then if for the rest of people who were over 25, they continued with 1400 milligram of Dunalimab. If they were between 11 to 25 centiloid, they reduced the dose to 700 milligram. Then again, they repeated the amyloid PET imaging in 52 weeks. Whoever was under 11 centiloid, they moved them to placebo, continued with placebo. And whoever had readings of between 11 to 25 centiloid twice, like at week 24 and week 52, they moved them also to placebo. So then they stopped the medication up to 76 weeks. Now, let's take a look at the results. This is about the amyloid removal from the brain by Dunalimab. In green, that's people who were on Dunalimab. In gray on the top, those are people who were on placebo. And as you can see, right at 24 weeks, at 6 months, 40% of people, they were negative. They became negative. It means that this medicine is so powerful that it was able to remove amyloid completely. 40% of people became amyloid negative. By week 52, 1 year, 60% of people became negative. And by the end of the trial, 68%. Now, I want to make sure that you understand, we're on the same page, that people here, 40% of people here at week 24, they stopped medicine and they went to placebo. And here, 60% of people at week 52, they stopped the medicine and they went on placebo. Now, let's take a look at the results. So here, their primary outcome was something called IDRS. and that showed 32% slowing on memory loss. This is the test that I just kind of mentioned, the primary outcome for the lecanemab and aducanumab, the CDR sum of boxes. Now here they had 23% slowing. So remember, for aducanumab, that was 22%. For lecanemab, that was 27%. For this one was 23%. But if you take a look at others, 39% slowing, 23% slowing, 21% slowing, very similar numbers, but they're all positive. Now, this was their phase 2 clinical trial. After this, they moved to their phase 3 clinical trial, which is Trailblazer 2. But before reviewing that, I wanted just to show you this slide, which is very, very interesting. So this is about amyloid plaque stabilization after 1 year of lecanemab, Dunalimab discontinuation. What you see here in blue, these are people who are on medication, of course, up to 24 weeks. And as you can see, the amount of amyloid drops comes to less than 11, which is very, very low here. And here at this point, they stop the medicine at week 24. These people are not on medicine. Even 1 year after discontinuation of the medicine, you can see that there is really not that much of increase in the amount of amyloid. Still, they are negative. They're very much negative. So they did a calculation, and the data suggests that it takes 14 years before plaque reaccumulates to the baseline level. What it means that we don't need to continue these medications forever. We may need to just give them enough medicine to remove the amyloid and then just watch them and go from there. Now, the phase 2 results showed that there is a rapid and robust decrease in amyloid plaque level, and it's sustained without significant reaccumulation over 1 year, and also greater amyloid plaque reduction associated with less cognitive decline. So there is a correlation, how much amyloid we can remove, how fast we can remove, and the amount of slowing down of memory loss. So the phase 3 of Dunalimab, Trailblazer 2, this is again randomized double-blind placebo-controlled trial, the best type of trials. Close to 1,200 participants with early stage Alzheimer's. These are all amyloid proven by CSF and PET scan. And again, in this study also, participants completed their course of treatment with Dunalimab once they reached a pre-specified level of amyloid plaque clearance, and that was 11 centiloid. So then they stopped medication. Here's just the overall result. This is the only thing that they shared. So they are, as you can see, the top part and lower part, they did divide people based on the amount of tau. Remember those neurofibrillary tangles? So they divided them to intermediate tau and then combined high and intermediate tau. So what you have here in the middle column here, and then right-hand side, 2 different columns. Those are 2 different statistical analysis. Here, that's mixed model repeated measures. That's a type of statistical analysis. And then they did natural cubic spline. That's another type of statistical analysis. And the reason that they put these 2, because they wanted to show that this is not about how we analyze the data. Whatever we do, the results are quite significant. And for those of you who know the p-value, the p-value is quite significant. The more zeros that you see here, the higher the significance of those p-values. But let's take a look at the numbers in terms of clinical efficacy. For the CDR sum of boxes, this is the one that I kind of mentioned about other studies, 36% slowing down. For the one that is about the function, 35% slowing down. So numbers are quite robust. This is very exciting. So essentially, as you can see, we have consistent results in all these 3 different anti-amyloid medications. Now, who is my ideal patient for anti-amyloid therapies? And of course, as you can imagine, in the clinic, there are not really too many ideal patients, but this would be someone that I would consider strongly for these medications. Of course, this is patient's decision, but these are patients that we will have that discussion about these kind of medications. People who are healthier with up to 85 years of age, and they have positive biomarkers for amyloid, either PET imaging or spinal tap. They have milder symptoms, early stage of Alzheimer's. There is less shrinkage on MRI when we do the MRI, and there is really no brain bleed in the brain, and they are not on blood thinners. And the reason for these two is that these medications increase the risk of bleeding. And also, very importantly, patients who are very much motivated and reliable and have strong support. When I say reliable, it means that they will show up for their follow-ups, because these kind of medications requires quite a few of surveillance MRIs and follow-ups. For those of you who have been exposed to these medications, I know that there are some people right now, you know how it is. So those are people that will be eligible for these kind of medications. Now, anti-amyloid immunotherapies, this is really the beginning of disease-modifying therapies. But as you can see, most probably the amount of slowing down would be something around like 40%, meaning 8 months gain within 18 months. So that's probably what we will get if we just target amyloid. So now, how to achieve bigger impact? That's a big question. Maybe we should explore anti-amyloid drugs before symptoms appear. So right now, when we, I would say, target these anti-amyloid medications for our patients, those are for people who are symptomatic. So what you see here is the continuum of Alzheimer's disease. And as you can see, starts with preclinical, and then of course people progress to symptomatic stage, the MCI or prodromal stage, and then dementia when they are becoming very much symptomatic. Right now, what we target is mainly the MCI and dementia stage. And at this stage, we get probably about 40% reduction or slowing in the amount of in the memory loss or functional loss. We may need to move a little bit earlier to the preclinical stage to hopefully get something close to 100%, meaning to be able to prevent Alzheimer's disease. So, where do we go from here? First of all, again, preclinical, when people have no symptoms of memory loss, that's a very viable option, and I'm going to describe and explain that. But the other thing is combination therapy, because as you can see, there are quite a few different mechanisms that will cause probably Alzheimer's or be contributing, as you can see here, all of those different circles are different mechanisms. And right now, with these medications, we only attack the amyloid plaques with anti-amyloid medications. Maybe to a certain point, we decrease the amount of tau. I didn't really show the evidence, but we do. By attacking amyloid, we can reduce the tau deposition, but there are quite a few different options that we have. Hopefully, we can increase that 40% to maybe 50%, 60% in symptomatic people. Now, I just wanted to show here about Alzheimer's disease spectrum. It's like this big iceberg. What we see in a memory clinic is really the tip of iceberg. People who have dementia and MCI, very mild memory problem, that they are symptomatic and they come to our clinic. But there's a huge part underneath, and these are 8 million people right now in the US over the age of 65. Essentially, 1 out of 3 people over the age of 65, they do have amyloid plaques in the brain, but they haven't become symptomatic yet. But based on evidence, they have enough time. Some of them will come to the surface, and maybe this is ample of opportunity for us to stop Alzheimer's or prevent Alzheimer's by intervening at the preclinical stage. And one of those studies is actually a AHEAD study. So the AHEAD study, it's a prevention clinical trial that is testing whether a new treatment, which is lecanemab, delays memory decline in people up to 20 years before symptoms appear. Now, you may ask, why preclinical AD? Why should we really go early? Well, first of all, it is now possible to accurately diagnose Alzheimer's at the preclinical stage prior to substantial irreversible damage to our brain, as I showed before, so we can detect people who have elevated brain amyloid plaques but no symptoms. And you can imagine intervention prior to dementia stage, which we think is irreversible brain cell loss, lots of shrinkage and atrophy and brain cell death. So intervention at early stage we may have better chance of changing clinical course. And think about other disease processes, what we do, for example, in patients with diabetes or HIV or cancer. Imagine if someone comes to me with colon cancer and I do my work and then I figure out that the colon cancer is confined to colon. And I would say, well, go home, please, have fun for 5 years and come back when it's spread out. What would be the response? I don't think that I can probably practice anymore. Tomorrow, my license is removed, right? That's not logical. That should be the case. That should be our approach in patients with memory issues as well. We really need to prevent rather than treat. So AHEAD study, this is 2 different clinical trials. So participants are enrolled based on the amount of amyloid plaques in their brain. The A3 trial, participants have intermediate level of amyloid plaques. We don't even consider them as positive. So this is the first ever trial that is very close to primary prevention of Alzheimer's disease. And they receive lecanemab every 4 weeks rather than every 2 weeks for 4 years. The A45 trial, participants have very much elevated level of amyloid plaques, and they receive the medicine every 2 weeks for 2 years. And then after that, when we are able to remove the plaques, then we go to every four weeks for the remaining of the study, which is another 2 years. So this is really a precision medicine. It's funded by National Institute of Health and several philanthropic organizations, and also Eisai. Eisai has the lecanemab. It is conducted and coordinated by the University of Southern California and Brigham and Women Hospital. There are 100 sites, 79 in North America, and Eastern Virginia Medical School Glennon Center is one of the active sites. We have screened more than 100 potential participants, and I think right now we have 7 participants, and within the next few weeks we will have more added to those numbers. So we are very excited having this trial available. Now, this is really a first of its kind to enroll participants as young as 55 years old who are at risk of developing Alzheimer's, but they haven't been diagnosed or they don't have symptoms of Alzheimer's. And it also targets the earliest changes in the brain due to Alzheimer's disease. As I said, we enrolled participants who have intermediate level of amyloid plaques in the brain. And it also tailors the dose, as I said, based on the amount of amyloid. We have 2 different dose regimens for this study. And of course, the medicine lecanemab has already been shown to slowing the memory loss in people who have symptoms of Alzheimer's disease. So this is quite exciting having this medicine right now for our prevention clinical trial. Who is eligible? Age of between 55 to 80, and they haven't been diagnosed with Alzheimer's, but they are worried about a memory loss in the future. For example, they have family history, or they have genetic predisposition, or even just they have some risk factors like vascular risk factors, and they want to be checked. And they agree to a 4-year commitment, and they have a partner with them that we need to probably talk once per year, I would say, to get some information about the participants' thinking process and functional status. How about planning future prevention trials? Well, already there is A2 study that is the design is ongoing. And this is about active vaccine aiming at amyloid. So this is now primary prevention. What it means that for people who even don't have the plaques in the brain, we give them a vaccine to immune against those plaques if they are going to build up. And of course, start at a very young age, 50 to 65, because at that time, the immune system is intact and there's less potential for side effects. And also utilize blood biomarkers to find patients. This is very exciting field right now, the blood biomarkers. We are very close to have something that we can do instead of just doing the spinal tap or doing the I would say the big and expensive test of PET scan to do blood test. In fact, we do have biomarkers discovery. Some of you probably know, and this is very promising. So, future trial design. This is mainly the future of the clinical, I would say, practice is combination therapy. Similar to what we do with HIV, diabetes, or cardiovascular problems. We start one medicine and add on second, third, fourth. So that would be combination of, for example, anti-amyloid medications, BACE inhibitors. That's another type of medicine aimed against amyloid, anti-tau medications, or inflammatory medications. So that's really the future of clinical trials and future of practice in Alzheimer's disease. Well, I'm going to stop there. This is our wonderful research team that I know that some of them are also right now present. And I also wanted to share with you that there is an Alzheimer's Association annual dementia conference, and that's on June 24th. Dr. Carl Hill, who is the Chief Diversity Officer from the Alzheimer's Association National Chapter, he will come here and we'll talk. He's a great speaker, and I will also talk there as well. So this is a great program. So if you are interested, the information is here. You can certainly register, and I think that will be a great opportunity for some of you. And I'm going to actually stop by just sharing our website of our research. We have quite a few different research studies going on, both clinical trials and diagnostics. And we're bringing, I think, 2 or 3 additional research studies within the next few months. One will be, again, Envision trial for the Aducanumab, which will hopefully will start next month. So we are very excited about that. So I'm going to stop sharing here, and I'll be more than happy to answer any questions that people may have.
Dr. Okhravi, this is Jim Morton.
Hi, Jim.
Hi, sir. How are you?
Doing very fine. Thanks for attending. Appreciate that.
You're welcome. I found the presentation very helpful. Very complex subject, to say the least, and you explained it beautifully. So thank you. My first question, I guess, is 3 major, I guess, disease-modifying treatments. As you know, we've been involved in the Alzheimer treatments for some time. But the question is one of the 3 drugs appearing to be better than the others, or is there a possibility of jumping, or should we be considered jumping to other treatments?
Very good question, and that's really open for discovery. We don't really know exactly. Just to give you a little bit of a background, as you know, and I mentioned for Aducanumab, for example, they stopped the clinical trials prematurely. So my thought is that if they allowed full clinical trial duration, probably they would have come up with very similar results that we had in Lecanemab and Dunalimab, but unfortunately, that's not the case. Hopefully, we will see that with the Envision trial, because this is going to be a complete clinical trial. Now, Lecanemab and Dunalimab and Aducanumab, there is one specific criteria or characteristic of these medications that 3 of them are able to reduce the amount of amyloid plaques to less than 25. And that seems to be the magic point. And if it comes to less than 25, I think the effect probably will be something between 20% to, I would say, 25% to 35%. And to be honest with you, 25% to 35%, there is really not that much of difference. Now, what is different is the approach that they had in Dunalimab, as I kind of explained, stopped the medicine after they removed the amyloid plaques. So that was not, of course, the case in Lecanemab trial or Aducanumab trial. But that could be also true for those two medications because they work very, very similar. However, because they didn't have that in their design, I don't think that we are going to change the protocol, for example, for Aducanumab or Lecanemab, to give it to the point that we stop when people have no amyloid plaques in the brain. Probably we need to continue for a couple of years, and then after that, repeat the amyloid PET imaging, and if there's no amyloid plaques in the brain, then stop and watch. Let's see, actually, in the chat, let me see if there's any. So if there is any question, please feel free to unmute and ask questions.
Hi, Dr. Okhravi. This is Madeline.
Hello.
Hello. I am wondering the use of the infusion versus injection versus pill form for these three medications.
Very good question. So these medications, there is really no right now pill form. However, there is subcutaneous sub-Q injections. For one of them, essentially, there is a trial that is going to start very soon. But at this point, we don't really have any evidence of efficacy comparing at least the subcutaneous versus the infusion. So at this point, the only way that these medications are available and are effective, we know that's infusion, but that's really the next step. In fact, there is another clinical trial that Eli Lilly is going to do. The medicine is very similar to Dunalimab, and in that clinical trial, and we are going to be a site, it will be sub-Q injection every 7 days. It's like very similar to medication that people give themselves for rheumatoid arthritis or other types of autoimmune disorders. So those are not available at this point. At this point, it's just only the infusion.
I'm trying to ask a question. How do you know? Hello?
Yes. Hi.
This is Ann Stensel. Ray Stensel's my husband.
Sure.
How do you know how or when or if you qualify for a trial?
So the two main criteria for qualification is, first of all, have early stage Alzheimer's, meaning that the memory loss is not significant.
Right.
It's very mild or mild. That's one of the main criteria. The second criteria is to have those plaques in the brain, because these medications are aimed against those plaques. So if you're going to give that to someone, that we better know that there is plaque in the brain, so then the medicine could be effective. Those are the main criteria, but there are, of course, quite a few other criteria as well. For example, age is important. The clinical trials, they have a set age, and as I said, for example, for Lecanemab, that was 50 to 90, and for Aducanumab, was kind of a little bit older, 65 to 85. So that's another major criteria, but there are quite a few different ones, and people who are interested, when they come to us, we check all of them, make sure that people are eligible, and there's really no ineligibility before they get enrolled into this study.
How do you find out if you've been enrolled or can be enrolled if you're positive for all of those things?
So I assume that the question is, how do you find out, right?
Yeah.
Well, I think the best way is to have that discussion, for example, with us, and then figure out whether you have the preliminary eligibility criteria, and if so, then we can move forward with the next step and then next step.
Thank you.
Sure.
Dr. Okhravi?
Yes.
I'm Dr. Butler. I am I to assume that on this study, you're again studying patients only that have plaques? You, as you know, I know you have for at least 4 patients that don't have plaques, but positive spinal fluid. Are they going to be involved in this study or not?
Yes, that's a very good question. Again, I know, Dr. Butler, we have had that discussion before. I believe the Envision trial, if I'm not wrong, they do accept people who are CSF positive. I'm not 100% sure about that, but the other study that we have, which is a subcutaneous medicine, in that study, CSF is acceptable. So essentially, it's either CSF or the amyloid imaging, amyloid PET imaging. Those are both acceptable.
Thank you.
Sure.
I have a question. Hi, my name is Catherine Finney. Can you hear me?
Yes, yes.
Hi, I have been diagnosed with mild cognitive disorder for a number of years. And I was wondering, I am 77 years old, and I was wondering if I would be eligible for a trial. I don't want to really get Alzheimer's if possible.
Sure. Well, I think we can certainly look into that and figure out if you're eligible for the study. Well, obviously, based on your age, you are eligible. But we probably need to look into your medical record and figure out if you'll be a good candidate. And of course, as I said, one of the main criteria is to have either the proof of amyloid plaques in the brain, either by CSF, spinal tap, or the amyloid PET imaging. But we do have studies that we can certainly enroll you into to have that, let's say, amyloid PET imaging, or just move forward, with spinal tap if you haven't done.
Yeah, I've had a spinal tap already.
You have had, and if the diagnosis was Alzheimer's, then you have a good chance that you may be eligible for the study. Yes.
Okay, well, the diagnosis was not Alzheimer's.
Then
Right now, it's just mild cognitive disorder.
Well, there was, when we met with you, I'm Joe, I'm Douglas's husband. When we met with you, you said it's not positive for Alzheimer's, but there's something not good going on in there.
got it.
Those were your words. And then we talked about the PET scan, that we might be eligible for a PET scan as the next step.
And are you right now in line for the PET scan?
We're just waiting to hear from you, because I understand there's a long wait.
Yeah. So if you're waiting, you're going to get a call from our research coordinator, and I think that will determine. Dr. Butler asked a very, very good question about discrepancy between the spinal tap and the amyloid PET imaging. About 10 or 15% of people, one could be positive, but the other one could be negative. So you may be one of those people that your CSF is negative, but your PET scan could be positive. So I think the best is just to wait to have that amyloid PET imaging. And then after that, if it is positive, then yes, you will be eligible for the study, at least based on preliminary results.
So how would I go about trying to register or something?
I think you're already on the list.
Okay.
We've already talked to Dr. Okhravi about it.
Okay.
You're going to be contacted by our team.
Yeah,
Okay.
we'll get an email
Okay.
if you watch your emails.
Dr. Okhravi, you have a couple of questions on the chat now.
Okay. I can probably read there. So let me see here. Can you comment on safety concerns, potential contraindications in patients on antiplatelet or systemic anticoagulation? It's a very, very good question. So I didn't talk about the side effects just because of the the time. So these medications, thanks, Dr. Bluestein, of bringing up that question. These medications have one major side effect, and that's called ARIA, amyloid-related imaging abnormality. And ARIA, there are two different forms of ARIA. One will be fluid leaking out of blood vessels in the brain, so that causes swelling. And the second one would be blood leaking out of the blood vessels, and that would be very small areas of bleeding, we call it microbleeds. Now, based on the Aducanumab trial, 75% of people who develop this abnormality, ARIA, they will have no symptoms whatsoever. 25% become symptomatic. Out of those 25%, only 6% they have to stop the medicine completely. And the symptoms are nausea, sometimes vomiting, headache, or speech issues, balance issues. Those are the main symptoms related to ARIA. And what we do when they happen, we actually stop the medicine. We repeat the MRI after a month or maybe two. And when it's stabilized, then we will restart the medicine. So majority of time, you're able to restart the medicine. So we know how to treat and we know how to manage those kind of symptoms. Before, about 15 years ago, that kind of looked very scary, but it's not as scary as we thought, it was before. We can manage that for sure. So that's the main side effect. Another side effect is about infusion reaction. Sometimes people may have some, drug reaction. For lecanemab, that's about 25% of people. For aducanumab, that's, less. But that usually happens within the first infusion. And for those people who develop symptoms, we give actually medicine for their second, third, or fourth infusion. So those are the main side effects. And for that reason, we do surveillance MRIs. There's a specific protocol that we do have, and we repeat those MRIs at specific time, make sure that we're not missing anything. Now, I see another question from Dr. Nesbitt. Any anti-inflammatory properties noted in any of the anti-amyloid therapies? Well, they do have anti-inflammatory effect in a way, because I would say, well, yes and no, because the way that they work, these anti-amyloid medications, they do engage microglia that are inflammatory cells in the brain. And that's the way that we get rid of those amyloid plaques in the brain. So from that standpoint, they actually provoke the immune system in a nice way to fight against those plaques. But amyloid plaques in and of itself, they can also, it can also increase inflammation in the brain. And I assume because they reduce the amount of amyloid plaques in the brain, so they may have some anti-inflammatory from that standpoint. But otherwise, they actually, I would say, stimulate our immune system with microglia, and then microglia is able to remove those amyloid plaques from the brain. And then another question: if the patient on the Envision trial, after it's finally launched, shows significant improvement with Aduhelm after several months, will those getting the placebo be offered the drug before the end of the trial? Yeah. So one thing that I wanted to make sure that we're all on the same page: these medications, they do not improve memory. They just slow down. So that's very, very important for you to kind of keep in mind. They do not improve memory. They slow down memory loss. Now, during the trial period, which is the 2-year period, everyone is going to be blinded. So no one will know whether people are on medicine or not. And after 2 years, when they open the label, if people are on medicine, they're going to be offered to stay on medicine. And if people are not on medicine, they're going to be offered to go on medicine. So after 2 years, yes, that's a possibility to continue with medicine if you're on medicine. And if you're not on medicine, go on medicine when they finish this study. I think that's as much as I can see in the chat. I don't know if there's any other questions. Seems that we really addressed all the questions. Madeline, I'm going to just turn it to you.
Thank you, Dr. Okhravi.
Thank you.
Thank you.
Thank you so much, Dr. Okhravi.
Thank you. Well,
Thank you.
I think we can conclude and have a wonderful day, everyone. Thanks for attending.
Thank you. Thank you.
Bye.
Night.
Thank you.
I said it.